For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 3, Episode 36 of Surfing the NASH Tsunami. Our look at the new release of Regenerate data last week starts now.
This week on Surfing the NASH Tsunami.
On July the 7th, 2022, Intercept pressed that they announced positive data in fibrosis due to NASH from a new analysis of its phase 3 Regenerate study of obeticholic acid.
I think this is a groundbreaking study for me. I think Zubair called it a watershed moment. It's for sure the first phase 3 trial that was positive. Looking at the numbers, I agree. It's the same effect size.
There has been a lot of consternation in the field that the FDA has set a bar that is, quote unquote, a moving target. The goalpost move. It's too hard to hit. It can't be done. Well, reading histopathology two distinctly different ways, obeticholic acid and Intercept have shown that it can be done again.
The bigger safety concern was whether the change in lipid profiles was going to mean anything at the end of the day. We've now got patients out 3 times as long as they were then. Nothing has changed, and if anything, the lipid issues seem to have somewhat resolved with longer periods of therapy.
The amount of data that we get about how it get people through the study is really important, and we very, very rarely see that delivered in any study, apart from the way it was designed, how the pathway was, and what the visit schedule was.
When I see a patient with elevated liver chemistry test and fatty liver, I don't ask, you have itching? Are you itching? Because inevitably, they start itching because you ask the question.
I'm just scratching my head now, right?
We have to give Intercept accolades here in the sense that they didn't give up. They persevered, they continued to drive forward, and they added 3 different adjudication committees.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researchers and key opinion leaders Dr. Stephen Harrison and Professor Jarn Schottenberg, liver wellness advocate Louise Campbell, and pricing and forecasting guru Roger Green, as they discuss Intercept Pharmaceuticals' press release updating on the Regenerate trial, this week on Surfing the NASH Tsunami.
So there are some weeks where figuring out what to do the episode about is really a challenge because there's nothing that current. And then there are weeks like this week where all kinds of heck broke loose last week. The Regenerate re-release came out. The nomenclature conference on what are we going to call this blessed disease was in Chicago for 2 days. And I wasn't there, but I've heard some stories already, and I'm sure we'll hear more before we're done. Stephen and Jorn both were there. So the nomenclature conference material is not ready for discussion yet because there's a lot left for folks to sort out. But Regenerate certainly is. So that's what the 4 of us are going to talk about today. We had hoped to have a couple more people with us, but nomenclature played heck with people's schedules, and everyone's doing catch-up today for all the work they didn't do on Thursday and Friday. Unless, like Jorn, they had to fly home overnight, and they're now in their, 42nd hour straight of being awake or something like that. So, Jorn, how are you feeling today?
Well, fairly good. I'm getting a little tired here, but it was a full day for me. So towards the end, changing time zones again, I'm looking forward to go to sleep and get a good night's rest.
Well, Stephen, your job is, as the guy who's going to do more of the talking today, your job is to keep Jorn energized.
I'll do the best I can. He's usually a ball of energy, so even getting him 2 or 3 notches down, I'm sure we're still going to be in for a treat today with Jorn.
I'm sure we will. And Louise, how are you today? We have to get this done on time because Louise has to watch the English women play the Norwegian women in the Euro 22 female.
Absolutely.
And Stephen's going to have people outside his door, and I'm not going to go into everything I got to do, but that's all right. So with that, why don't we just dive into Icebreaker? One good thing that happened last week, personal or professional. Take your pick, brave one, go first.
Oh, I'll jump in. Having recovered from COVID, spending 3 lovely days in the Reims, I don't think that's how it's pronounced, but the Champagne section of France, doing the one thing I really enjoy, tasting champagnes. I have bubbles on a Friday. That's my motto, and I live by it. So tasting some new ones and some recommended champagnes was lovely, and it's nice to get away, and the weather was very much like this.
So we'll have to discuss when you're sponsoring the champagne tasting for everybody, but this sounds promising. I like it.
Yeah, picked up a fair few bottles.
So you do Bubble Friday, and then for Mundane Monday, you do the podcast. So it sounds like 4 of the 7 days of the week are taken care of.
Absolutely. but only one of them's alcohol related. So that's a benefit.
That's true, because this is non-alcoholic fatty liver disease. There you go. Very good.
Well, I can't other than say, of course, the nomenclature conference was a professional highlight. I was moderating one of the breakout sessions, and it was good engagement. I think many colleagues debating different aspects of the disease, patient advocates. So clearly that was very exciting. And it also has a very personal side to it. So meeting a lot of friends and being invited over to Renella's house at night. And that was a professional and a personal highlight. The one thing I'm not going to give away on this session is, of course, there was a bartender that prepared special drinks, and that was another highlight. I'm going to share that with a group at one point in the future.
We might decide to post pictures on the website. I've seen the pictures. I think they're brilliant. Stephen, you're up.
I participated in the nomenclature conference virtually because I was in the middle of moving my 21-year-old son out of an apartment into a house they're renting for his senior year of college. And just being able to spend time with him was terrific. Although we did comment that, I have a big truck, an F-250, a Ford F-250. It took 4 loads to get his personal stuff out of that apartment and into a house, of which I wanted to take 3 of the loads and throw it away. But we didn't do that. But the highlight was just being able to spend time with him. And, we don't get to see him that much anymore. So the older he gets, the smarter he gets. He realizes that his parents aren't as dumb as they once were. And we're growing up in a hurry in front of his eyes. Our intelligence is starting to come back. So, we can actually have good conversations now, and just a fun weekend with him, and popping into the conference as much as I could in between as well. I thought Jorn did a masterful job moderating a couple of the virtual sessions, and it was great to hear the debate that occurred. And I'm sure we'll have further discussions about that as we roll along in a future podcast. But anyway, that was my highlight for the weekend.
If we can get all that into one podcast, Stephen, I'll be really impressed. My money's on it's going to take multiple podcasts over time to tell that whole story. At any rate, I just had a really quiet weekend. Nobody was sick. The weather was perfectly temperate, and I actually got to kick back a little bit, which I never get to do. So just take that in its own right. Not dramatic, but really fun. And I didn't have to get on an airplane, so I didn't have to worry about any of those things. With that, why don't we just get started? This podcast actually, the first time it started showing up 2 years ago on charts of how many listeners people get, was the week that FDA issued the original complete response letter in response to the Regenerate data, which is about 2 years ago, or after last week. It was right at the end of June. 2 years later, like clockwork, here we go. Stephen had been talking about this a couple of months ago, that they've been relooking the data, and they've had people on it for over 4 years and all the things that were in the press release. And I guess the real question is, they've put this data out, so what's new and what do we think it means? We're fortunate to have Stephen with us today, because actually there's no one I'd rather have answer that question if I only had one person answer it. So Stephen, why don't you walk us through a little bit what's in the press release, and then we'll stop as we go along and talk about what it means.
Sure. As we highlighted along the way in one podcast, there were a couple potential drivers of where the NASH field was going to go in 2022 and beyond. And one of the drivers that we highlighted was a potential for obeticholic acid to come back into play and be in the news and be a potential driver, or at least provide some horsepower to the engine of drug development in NASH as we move forward. So, with that being said, on July the 7th, 2022, Intercept pressed that they announced positive data in fibrosis due to NASH from a new analysis of its phase 3 Regenerate study of obeticholic acid. And there were 4 top-line bullet points that Intercept wanted to make. And I'm just going to read them, and then we'll talk about specifically some of the data. Number 1, the 25 milligram dose of obeticholic acid met the agreed primary endpoint of improvement in liver fibrosis without worsening of NASH at 18 months. And the p-value was highly significant. It was less than 0.0001. For those of you that aren't counting, that's 3 zeros and a 1. And that was consistent with the original Regenerate analysis. Now, I'll get into that in a minute. Point 2, the 25 milligram dose of obeticholic acid demonstrated double the response rate in reduction of liver fibrosis without worsening of NASH versus placebo. We'll get into that in a minute. Number 3, this data set includes larger and more robust safety database of 2,477 patients with nearly 1,000 on study drug for 4 years. Number 4, Intercept to resubmit a new drug application. So they're going to refile an NDA in liver fibrosis due to NASH. A pre-submission meeting with the FDA is scheduled later this month. So there you have it. That's their main bullet points. Now, just to dive into this a little bit, and then I want to, maybe we'll take this section by section. The first one is this idea that the 25 milligram, the high dose of obeticholic acid, met the primary endpoint, and they say consistent with the original Regenerate analysis. Now, here, they did a new interim analysis of the intention to treat population from Regenerate. This was the 931 patients originally submitted, underwent a new interim analysis. And I'll talk about what that means in a minute. But now, 22.4% of the OCA 25 milligram cohort met the primary endpoint of at least one stage improvement in fibrosis without worsening of NASH at month 18 on liver biopsy, compared to 9.6% of subjects on placebo. Now, if you remember back to the original data that was submitted, that 22.4% is not far off from the top end of 25. I think it was like 23% or something like that. And we can dig into the specifics, Roger. You may know them off the top of your head. But what happened was the placebo response rate went down a bit on reanalysis, 9.6%, giving you about a 1%, maybe a little more, increased treatment effect delta when this data was reanalyzed. Now, what was done different? Well, the population in this new interim analysis, that 931 patients, mirrored the original analysis population from 2019, but they used a different approach to read the histology. So instead of relying on two readers reading half of the data each independently, and now they used a consensus panel approach. And they're very quick to point out that this new consensus panel approach was in line with recent FDA guidance. So here, they didn't just take the word of one reader. They had to be consensus on what the results were. And we'll talk about that more in a minute. And ultimately, the data is the data. A numerically greater proportion of individuals also had improvement in NASH resolution, but consistent with the original analysis, it didn't reach stat sig here either. However, you only had to have one or the other to meet the primary endpoint. It wasn't a co-primary. Co-primary means you have to hit both. It was just a dual primary, meaning you had to have one or the other. And in this case, they hit fibrosis with a treatment effect delta that, I mean, let's be honest, it's really about the same as before, one or at most maybe 2 percentage points better, mainly driven by a lower placebo response rate. So let me stop there and let's have a discussion about this, because does that, in your mind, again, understanding the therapeutic index of a drug is driven by its efficacy. But as we learned from the original complete response letter and from many other drugs that the FDA has evaluated, that safety and tolerability are critically important here as well and play a big role in the therapeutic index. So all I want to focus on here is not safety and tolerability for the purpose of this initial point. It's just efficacy. And I want to get your perspective on this. Does this really change our perception of OCA in NASH, for one? And number two, does this change our perception of this drug as a driver of the field in drug development for NASH? And I'll hold my comments till I hear from everybody else, but maybe I'll open the floor up to discussion on this point.
Since I'm always the one who says brave one, go first, except when we're doing International NASH Day and Jeff McIntyre's moderating, I never get to say, okay, I'll be brave. Stephen, I think is exactly the right question on the efficacy side. And to me, the answer is nothing changed. They had stat sig at the higher dose last time. They have it this time. It was in the low 20s last time. It's the low 20s this time. I was struck that placebo dropped a little bit. I'm mindful of the idea that came out of the Q ballooning work, that you get people who consistently overread and underread. And my hypothesis has always been that if you go to consensus and you do it right, overreading is what's going to filter out. And that appears to be, well, not that there was a lot of overreading here, but that appears to be what happened here. Placebo went down, product held about where it was. I didn't see any change on the efficacy side of the equation from where we were a couple of years ago, number one. But number two, I don't think that this makes FXR a major reference for drug development around it, in part because it's not going to have 3 years in the market on its own, which it would have in the first place, and in part because we're now looking at a lot of drugs that appear to have better effect and quicker. So it may have a place, it may have value, but I don't think we're going to see people rushing out to figure out how to be the second drug in behind OCA. That commercially wouldn't make any sense to me.
Sure.
Let me follow up. I think this is a groundbreaking study for me. It was published in The Lancet in December 2019. It was the first phase 3 trial that showed that we can meet the primary endpoint of significant reduction in fibrosis compared to placebo in 18 months with paired biopsies. So this is, I think Zubair called it a watershed moment. It's for sure the first phase 3 trial that was positive. And looking at the numbers, I agree, it's the same effect size, but in the end, it's significant at that time frame. And as such, it reminds us that FXRs do have, according to this data, an effect here in NASH in being able to improve fibrosis. And I think it'll be important to discuss what was different in the type of reading that was applied here. And from my understanding, what's been written in the press release is that it's a little closer to what regulators would suggest on how to do it. But I think you have more insight on that, Stephen, in a moment. I think for the company, there's been a lot of talking about, well, it's only 11% difference delta. It's not much higher in this type of reading. I'm not so disappointed by the absolute difference between placebo, but it's the fact that it was actually a positive trial that still makes me say this is very important for them.
Yeah, and I think I'll echo what Jean said there, but I think there's also the aspect that if you look at it, yes, it hasn't changed too much, but we're 2 years down the line. If we approve it on this marginal difference, did we miss an opportunity 2 years ago to affect people's lives positively by putting this drug through to patients? And this has been the concern. It was the concern, I think, when we discussed it with Donna, that it was disappointing that they were asking for more. They now have more, arguably just a little bit more. However, that 2 years, has it really changed the ball game? Too much. But it will probably get approval on that. So we are talking about minuscule alterations, but that's a lot of people that were affected negatively in this past 2 years, and whilst we wait for this approval to come through.
So before we go talk to approval, and I think you're addressing the most important part that's actually in that press release that we haven't discussed, and I think Stephen's going to follow up on that. There is significantly more data on safety now, and I think that's the real game changer here in this press release, and we'll get to that in a moment, I think.
Yeah, we'll get there. I reserved my comments until I wanted to hear yours, and I think they're all very important. But let me just foot stomp what Jorn said. I do think this is a watershed moment that is kind of reinvigorated from 2 years ago in this regard. There has been a lot of consternation in the field that the FDA has set a bar that is, quote unquote, a moving target. The goalpost move. It's too hard to hit. It can't be done. Well, reading histopathology two distinctly different ways, obeticholic acid and Intercept, have shown that it can be done again. So I think this should put to bed the notion that the endpoint, at least of fibrosis improvement, is a bridge too far. It's a hurdle that's too big. It can't be done. Yes, it can be done. We could debate the treatment effect delta for fibrosis, and is that meaningful? What I would say is let's don't be myopic here, because what we haven't seen yet from this data set is how much we're preventing progression of fibrosis as well. And as Jorn and I will tell you in clinic, if we have an F3 patient that we can keep an F3 patient and prevent from developing F4 disease, more likely than not, that patient is completely asymptomatic, leading a normal life. And if we can keep them there, great. Let's manage his comorbidities and talk about a drug that can help us with that as well. So I don't want to throw the baby out with the bathwater focused only on improvement of fibrosis. I think just as importantly, there should be a conversation around, in this large number of patients, are we preventing progression of disease? What I'm a little challenged by, and this is probably just my naivety, and look, I don't mind throwing my lack of awareness on this out into the open, and I'm sure somebody will point out the obvious, but I wonder, a significantly larger number of patients made it through the 18-month biopsy time point. And why couldn't they include the additional patients in a refiling? instead of just looking back at the same patients and reading them a separate way, why couldn't they have added as many additional patients that had made it to month 18 into this analysis? Because obviously that would have increased the power. Now, maybe there's an obvious answer I'm just not privy to. Let me address the comment about the methodology of the read. So I commented already how they did it the first time. 2 pathologists, each given roughly half the biopsies, and said, go give me the answer. And so that wasn't consensus. It was essentially one pathologist reading the pre and the post in half the cases, and another pathologist reading the pre and the post in half the cases, and then putting their data together. Now, if you look back to data that was presented in poster form on the methodology, and I'm reading straight off the poster, the flow diagram for methodology. In stage 1, each pathologist, and there were a total of 6 board-certified NASH-trained pathologists who underwent proficiency testing for this scoring system. They were allocated to 2 separate panels, Panel A and Panel B. So in stage 1, there was an independent read. Each of the 3 pathologists from a panel independently read 4 slides per patient, H&E trichrome at baseline, H&E trichrome at month 18 to score fibrosis stage, inflammation, ballooning, and steatosis. Then 2 of the 3 pathologists match on a score for relative parameter, and then that consensus score is recorded. Alternatively, if 2 of the 3 pathologists do not match, the slide images are read jointly by an entire panel. Joint agreement is reached on the score for a relative parameter. Yes, then that consensus score is recorded. If no, the panel deems the slide to be non-evaluable for a relevant parameter and no value is recorded. So that's the methodology here. You had 2 pathologists, or each pathologist would read the slides, and if 2 out of the 3 matched, you're good. If not, then all 3 pathologists came together and read the slide. If joint agreement could be reached, you're good. If still no joint agreement could be reached, the slide was deemed non-evaluable. Now, I don't know, because it's not mentioned, how many slides were deemed non-evaluable, but at least now you know the way this consensus reading supposedly took place. Now, all I did here was go back to the poster that Arun presented entitled Minimizing Variability and Increasing Concordance for NASH Histologic Scoring in NASH Clinical Trials. It was a poster 1620 presented at AASLD last year, 2021. So that's the way the methodology was done to my knowledge. So that's where we are. Any thoughts on that, or should we go to safety?
No, let me reflect quickly. I think it's important that we decide on what's shown in the biopsy slide, right? We do the biopsy, we want a clear shot answer. We can't get an answer from one pathologist saying this is F3 and the other one says this is F1. This is something that bothers me. And I think, as you highlighted, moving into a consensus, having experts discuss this, saying, have a look here, maybe you didn't take this section into account, or, how do you evaluate this? I think this is how you do it in clinic. You got a tough case, you come up to a colleague, you say, hey, have a look, these are the labs, what do you think? You reach a consensus. So this is how I would do it in my clinic with a difficult case. I think this is how it has to be done in reads here, and it should reduce the noise. And I think the data shows that too. The question I'm not aware is, how do the regulators view this? You've been involved in some of those discussions. And the second thing that came up, I think this is now digitalized slides.
That's right. Absolutely. And I agree with you, Jorn. This is exactly how it should be done. This is how we would do it in clinic in a tough case. And I think the regulators, to my knowledge, are open to this idea of consensus reading. I'll take it one step further. I think the regulators are open to assistance from AI digital pathology as well. Now, AI digital pathology, let's make it clear, is not replacing the pathologist. But I think it could be used as a companion diagnostic if you're able to have a conversation with the agency and talk about how you want to adjudicate your histopathology in your statistical analysis plan. So if you say, hey, look, we're going to use consensus, but we're also going to have AI digital pathology there as a companion diagnostic, meaning the AI digital path report is sitting there for the pathologist to look at as they adjudicate the histopathology slide directly. I think that the agency, and I don't want to put words in their mouth, but I think they would be receptive to having a conversation about that. And for sure, having a conversation about consensus read. Now, let's also be clear, AI digital pathology was not involved in the reporting of the data you see in this press release. This was simply a consensus panel approach to histology. Okay, any other thoughts?
Yeah, actually, I have one, maybe two. The second one, actually, Stephen, came from your last set of comments. The first one was I've just been playing in my head with why a consensus read would have the impact of taking placebo down and not having an effect on anything else. It didn't take it down significantly necessarily, but it took it down a bunch, a noticeable amount. And I'm wondering why consensus would do that. I don't have an answer off the top of my head. I spent a little bit of time noodling this around over the weekend and couldn't get to an answer that I liked. That's number one. Number two, which comes out of your comment and I think is more important, is it would be fascinating to see, as you point out, the degree to which this stopped progression. Because if this becomes an antifibrotic that 1 time out of 4 actually gets you down a level and 3 times out of 4 stops the progression, it makes it a pretty valuable agent in combination therapy. Not the backbone necessarily of combination therapy, but a valuable agent to have. Because at the end of the day, fibrosis is really what we focus on more in terms of patients' health. And that would be pretty compelling, I think.
Yeah, and I think just to add to that, I think as a patient, they want to know that their slides are being read by the best of the best, and that where there's any adjudication that needs, that we're going to go that extra mile to do that. And I think that's what's happened with the consensus. I think that was a positive move. I think we're seeing a benefit in this study, and therefore hopefully we'll see a benefit in other studies. But I would like to see AI in the background. We've now moved very far with AI, and it's there to help the pathologists. It's there to make sure there's nothing that's unreadable. And it would show the strength of histopathologists and their skill in reading, particularly with the consensus, if we just had that additional metrics, even if it wasn't used to approve the drug, but actually to prove that the whole system works. I think personally we can benefit from that. It's great to know that these slides are now being better read and getting positive data.
Yeah, and just to add to that, I guess it was kind of a coming out party at EASL in the sense that we began to show that drugs that are having an impact on liver disease in the setting of NASH are potentially also improving liver volume. One of the presentations I gave from Madrigal in the Maestro-NAFLD-1 trial was this reduction in liver volume. And then in the cirrhotic cohort of the open label trial, the cirrhotic open-label cohort of the Maestro-NAFLD-1 trial, in addition to liver volume reduction, there was spleen volume reduction and an inverse relationship to rise in platelet count. Now, I don't know what all that means necessarily. It sounds positive, but I think we have more work to do. But one of the things that we drilled down on in another oral presentation was using AI digital pathology. We were able to apply a correction factor for reduction in liver volume. And that allowed for actually an accentuation of the antifibrotic response seen with resmetirom. And I'm just speculating here. We can do that on the podcast. And so I'm speculating that there probably was a treatment effect on liver volume with obeticholic acid. Now, it wasn't measured to my knowledge, and we don't have liver volumes, but I wonder as we move forward if that should not be looked at more holistically across trials, because, maybe there's an even more accentuated treatment effect delta on fibrosis if you're able to apply that. Pathologists are doing the best they can. They're reporting what they're seeing. But if you have compressed collagen, particularly in zone 2, the data that we reported suggests that pathologists really can't see that change to the naked eye. We're going to learn more and more about this, but let's move on and talk about safety, because Jorn mentioned it. So safety was evaluated in almost 2,500 subjects who took at least one dose of study drug. So placebo, OCA 10, OCA 25. Compared to the original analysis, the safety population in this new interim analysis had significantly longer exposure to study drug, median 42 months versus 15 months previously, yielding more than 8,000 total patient years and 3.4 times more exposure. Nearly 1,000 subjects have been on study drug for 4 years. Now, so that's your kind of foundation. This is how much data has been collected. So what is the data? Treatment emergent adverse events, treatment emergent serious adverse events, and deaths were generally balanced across the OCA and placebo treatment groups. The most common treatment emergent adverse event, as expected, that was my editorial, was pruritus, 24% in placebo, 33% in OCA 10, and 55% in OCA 25. And pruritus was the most common cause for treatment discontinuation. Serious gallbladder-related events occurred in less than 3% of subjects in any treatment group, and consistent with its known mechanism of action, OCA 25 had higher rates of biliary events, including gallstones. Let me stop there because there is another section that talks about independent groups of experts reviewed safety. But before we get there, maybe just some comments on what you heard as far as the volume of safety data and what we know relative to pruritus, discontinuations, and gallbladder-related events.
So I think also the safety data is somewhat recapitulated, but of course the reassuring aspect here is you have much broader database. You have many more patients. There's nothing new coming up. There is no unexpected long-term detrimental effect from giving an FXR agonist in these patients. And the pruritus the numbers are, I think they went up a little bit by 1 or 2% compared to the original paper. So fairly comparable. The gallstone event rate, that was also known. It's just, for me, the robustness of the data has increased significantly, and considering the risk-benefit ratio that was always addressed as one potential reason for a complete response letter, I think here the data has just been improved by a lot.
I think it's reassuring with the increased amount of data and the volume. Given that PBC, I think the dose is 5 milligrams, and then you increase to 10 and the maximum dose is 10, to actually see it get to 25 milligrams and not cause overly concerning side effects and larger numbers is greatly reassuring in its previous setting and its approved mechanism so far. That helps from my perspective. If I'm engaging with a patient on this medication, then I think we should be already planning pathways into delivery of this, because just putting a patient on this type of medication with 55-52% pruritus is not going to be something you can just dish out in a medical practice and hope that they're going to stay on your medication. We have seen it be the biggest cause of withdrawals. And Roger, your testament to how pruritus can really cause problems, and we know it causes problems. So irrespective of the gallbladder and the gallstone, but do we have to add those as costs to this medication? This is not a cheap medication, even on its current approvals. So that's where it causes me slight concern, but the reassurance of the data is certainly, I think most patients will be really pleased to be able to be offered something that now has robust data on its safety and efficacy.
I was just going to foot stomp what Jorn had said and then maybe add some color to Louise's comments. So first of all, nothing surprising in what we've talked about so far. Again, consistent with mechanism of action, consistent with previous data. And there's maybe Jorn always has great insight, but one of the more important insights he made relative to this is that nothing new. So let that sink in. 1,000 subjects studied for 4 years, nothing new popping up. That, I think, is critically important.
Can I run with nothing new for a minute?
Yeah.
If I recall, and I was just getting involved with this disease when all this happened the first time, so I'm not going to claim my recollection is perfect, but the bigger safety concern was whether the change in lipid profiles was going to mean anything at the end of the day. And we've now got patients out 3 times as long as they were then, and nothing has changed. And if anything, the lipid issues seem to have somewhat resolved with longer periods of therapy. So nothing new takes on, if you will, new and exciting meaning, just to use the word new 2 different ways in the same sentence, when you keep in mind that the concern was what would happen in a longer course of therapy, and that didn't happen.
Well, for sure. Now, I think we need to see more data necessarily. I mean, this is a press release. There's only so much you can put in it. There is the paragraph in here that says independent groups of experts reviewed certain categories of safety events to provide a blinded adjudication as specifically requested by the FDA. Their focus was on hepatic safety, cardiovascular, and renal. Now, the next sentence is top-line analysis through 4 years of treatment showed a numerically higher number of adjudicated hepatic safety events for OCA 25. The vast majority were mild in severity. But I think we need more clarity around that. We need to understand what that means exactly. Because remember, the treatment effect delta is 11 to 12%. So how many people do we need to treat to have a benefit? And again, this doesn't take into account what I said earlier about preventing progression of disease. But that does come into the equation when we talk about therapeutic index. And so I think when we I see numerically higher number of adjudicated hepatic safety events. The vast majority were mild in severity. Well, read between the lines. The vast majority were mild in severity, but what about the rest? I think we just need to know more information. And then for adjudicated core major adverse cardiac events and adjudicated acute kidney injury events, frequency of events was low and balanced. Okay, that's encouraging. Very encouraging, because that gets to the LDL comment you made, particularly for the cardiovascular events, and it shows that OCA treatment was associated with an increase in LDL at month 1, which returned to near baseline value by month 12. Now, that's encouraging. Does that mean they got put on statins or PCSK9s or just kind of resolved on its own? I don't know. We probably want to learn more about that. Changes in other blood chemistries, including alkaline phosphatase, gamma GT, AST, and ALT, all showed evidence of FXR-mediated activity supporting the dose response of OCA. So again, that's encouraging. Let me just ask this. Jorn, when you read this, you look it over. I know you're encouraged by the efficacy, the consistency in the efficacy. You're encouraged by the overall totality of the safety data. Do you have any ongoing, lingering questions that you would like to learn more about based on what you read here?
There's a lot of things that come to mind, obviously. I think you very nicely detailed how the reading was done. That was my first question when I read that. And when people go to the press release, they will say, well, what was done differently now? And I think you really brought some light into that. That was my first impression. I was also looking for the LDL, and there is a comment on the LDL. I think you also mentioned that LDL actually returns near to baseline at month 12 in that larger data set and cardiac safety. So that was important. I think we've seen some safety warnings with cirrhotic patients in OCA and the PBC indication, and there's nothing coming up here. So that's another thing where, again, we're not hearing anything on that safety side. And what else would I be interested to learn? I think, I'd like to learn a little bit more of the other non-invasive tests. Are they congruent in terms of development? What happens to TE? How the transaminase is moving and how's that pair? I think we saw most of that, but just to be reminded, I'd probably go back and look in the 2019 paper, how that all looked like.
Yeah, I think those are important. Luis, let me get to you. I did have one thought just to follow up on Jorn's that I want to bounce off of him or everybody. What about the combination of NASH resolution and fibrosis improvement? How important is that for this drug to show that? Obviously it's a tougher endpoint to hit, but it's not commented on here.
That's a good point. And I think that wasn't shown also for the negative effects on the NASH resolution overall. I think the main message is this is a drug that affects fibrosis in this study, and I would be not expecting anything beyond that.
I think for me, it begs to ask about NASH and how the patient's diet altered or didn't alter. Because if you're going to eat the same thing, you're potentially going to continue the NASH spectrum, and depending on somebody's genetics. So that's the way I understand it. And maybe that's a different question. They shouldn't both be in the same outcomes, because this is a drug that affects fibrosis, therefore it ticked that box. Is it a drug that, or looking at placebo, or looking at sub-analyzing somebody's diet to see how that affected what was going on in the NASH? I think may well give me more information on that. When you were asking Jorn about what you would want to see more of, I want to know how many patients were counselled to stay on the study with pruritus. What were they given to reduce their pruritus? How intensive is the support to keep people on a study with pruritus? Because that is really vital when it comes to delivery of this drug in the real world. Because I've counselled people to stay on studies for hepatitis C when we were doing interferon or boceprevir, telaprevir. It can be hard work. It is a lot of input, but these people genuinely want to be there. So the amount of data that we get about how it get people through the study is really important. And we very, very rarely see that delivered in any study, apart from the way it was designed, how the pathway was, and what the visit schedule was. We don't get the additional muscle around, and I suppose the fat on the tissue around the skeleton of the study. That, to me, for any pathway design in any drug, is really, really important, but you don't see it. And in these drugs, it's going to be vital to know the breakdown and the intensity that was used.
So I had two thoughts. Stephen, given how little NASH improvement they had without worsening of fibrosis, was 6%, 6.5%, do I remember correctly? I just don't think there could have been an awful lot of NASH improvement, right? I mean, that's one patient out of 17. if, in fact, every patient that had an improvement in their NAS score with no worsening of fibrosis had an improvement of fibrosis, that would only be a quarter of the patients that had an improvement of fibrosis. So that 6.5 number was a big flashing thing that said to me, good for them for saying we're only going for an indication for fibrosis, because the NAS number just didn't seem to me to help them very much. Didn't hurt them, which is fine, but it didn't seem to help them.
Well, that's the other thing. We don't have a breakdown of the NAS here either.
Except that one reported number, right? 6.5 versus whatever it was.
Well, that's
placebo.
just NASH.
That's just NASH. Okay, that's right.
The other thing is they didn't report the Gestalt diagnosis of NASH resolution. That was the histopathologic required criteria of ballooning 0, inflammation 0 or 1. But definitely good point. Good point. I mean, when you look at combining the two, you wouldn't expect it to be very high just because the overall NASH resolution numbers are, around 6% or so, like you mentioned.
Stephen, and more broadly, the flavor I got, we're not ready to sum up yet, but I'll do a semi-sum up on this one point. The flavor I got from the press release and what they chose to analyze and what they chose not to analyze was the most conservative commercial approach you could have taken, which is okay, right? Remembering that we don't know what was in that CRL letter. It's never been revealed publicly. What they seem to have done is made some decisions or assessments about what in the CRL level letter gave them problems, and shared data targeted at minimizing or resolving those issues and not providing anything else they didn't have to. So if the presumption 2 years ago was that the efficacy numbers were okay, but it was therapeutic index or risk-benefit trade-off that was the challenge, then what they needed to do really was bring down the sense of risk. And these data are presented, I think, in a way that's designed to do that. How effectively it does that, different conversation, but that's what it's presented to do.
I think we have to give Intercept accolades here in the sense that they didn't give up. They persevered, they continued to drive forward, and they added 3 different adjudication committees, right? There's a separate renal, a separate cardiovascular, a separate hepatic, really trying to get into the weeds of what's happening here. And I think time will allow for a more robust, detailed introspection of what those results show. But at top line, I think it's positive. I would want nothing more than for this drug to become FDA approved to treat NASH. It's a great first start. It opens up opportunities for others to be advanced quite further along in the pipeline. I mean, as I said at the top of the hour, there's a real sense that there's a challenge in getting drugs approved for this indication because of the endpoint that the FDA currently has. And if we're able to get over that hurdle, even one time, the initial time, that would invigorate the field. And drugs that show significant promise would then potentially get the funding they need to continue in the pipeline of drug development. And first of all, I'm thrilled that they're going to submit a new drug application, and I'm hopeful that it'll be met positively. I'm also hopeful that soon we'll have more detailed analysis of not only efficacy, but of what these events are that have had to be adjudicated. And then I think, we'll see what happens with the rest of the field. But I have to look at the data that came out on July 7th as a very, very positive implication for the field. I'm hopeful that they'll get a positive readout from the FDA, and we'll see where it goes. But all told, I give it 2 thumbs up.
Please don't construe my comment as not agreeing with everything you just said. I do. All right. How they managed to present the data in the press release is simply tactical. But I agree with you. Look, they didn't fold up their tents. They didn't go home. They broadened what they did. People talk about throwing good money in after bad. Well, they threw good money in believing that it wasn't after bad.
Yeah, and I remembered a comment that Louise made about pruritus. So you're absolutely right. I mean, pruritus is nothing to walk away from. It's critically important. Patients don't tend to stay on things that cause pruritus. Pruritus in its severe form can be really psychologically damaging to patients. But remember, the way pruritus was defined in the study, it kind of is not the way we define it in clinical practice. In other words, Bjorn and I don't typically see patients and say, do you have itching? Unless they're a PBC patient or a PSC patient walking in the door and we're trying to make the diagnosis of an isolated elevated alkaline phosphatase, I'll say, do you have a lot of fatigue? Do do you have itching? Because those are the 1 and 2 common symptoms related to PBC. But when I see a patient with elevated liver chemistry test and fatty liver, I don't ask, you have itching? Are you itching? Because inevitably, they start itching. I mean, because you asked the question.
I'm just scratching my head now, right?
Yeah, everybody's itching now on the show. So that's what you had to do in the trial, is you actually had to ask them about itching, pruritus. And so whether that led to an increased reporting of pruritus, I can't tell you for certain that it did. But just like we all started scratching, I mean, there's something to that for sure. Now, I think that's where the drug discontinuation is a little bit more true. Although if I recall from the trial, there was a certain point at which you had to discontinue drug if there was a certain amount of pruritus. So again, to me, yes, that is something to follow. Dose titration might help mitigate that in some way, but I am really more focused on the hepatic events. And one thing we didn't talk about today was the cirrhotic trial, the reverse trial. That's complete. We haven't seen results from that. I think it would be incredibly important to have the safety tolerability profile of those 900 patients, in addition, because I think that will help us. Because, again, the first FDA-approved treatment for this disease, I don't know about you, but there's going to be a lot of well-compensated cirrhotics that are going to want to get put on treatment.
Uh-huh. No, I agree. And, that's going to make this a pretty complete data set. It'll be important to be reported out soon, hopefully. And, regarding your comment on the pruritus, I'm convinced there was a high pruritus report in the placebo group. There's something on how you ask and how it's reported that's critical. There will be some discontinuations related to it, but I think, as you rightfully said, to be taken that first barrier is going to be so important. And with this data here, in particular with the safety data and the extended data set on long-term exposure, raising no new significant issues, this should be revisited, and I'll be very excited to see what comes out of it.
We agree. So I have one experience I don't think any of you have had. Louise referenced this briefly. I have a couple of times on the podcast, which is I've actually discontinued a drug because of pruritus. And by the way, it was a cancer drug. And they were trying to see how many doses they could limp me through. And I finally said, forget it. I just can't do this anymore. But to get to that point, I had to have severe itching over, I'm going to guess 70% of my body 16 hours of the day, at which point I said, listen, I can't do this. I have a hard time envisioning that you would not approve this drug simply because of the pruritus issue. I mean, that's a controllable toxicity, right?
Yeah, I agree. It would be one thing is you stopped the medicine and the itching didn't go away.
Yeah, and it'd be one thing if you put a ding in somebody's cardiovascular system in a way that was going to have long-term effects, and you did that often enough. But I can't see it with pruritus.
I think at the end of the day, there are going to be, I'm hopeful that this gets approved second time around. My guess is if you had to hold my feet to the fire, there'll probably be some left and right boundaries around appropriate follow-up of these patients, just kind of like we do at PBC. I think the story is not told until we see the cirrhosis safety and tolerability data.
Yeah, and I agree, and it's about the planning. But if we're not planning now to roll out, because I think it's really exciting, this is one disease area that I just get excited that once these medications go into the real world, we'll actually get better results, because not only you cannot currently, under any trial, place in behavioural therapy into that trial because it would skew the results, quite rightly. But the minute you get to the real world, you have support, you have dietary discussions, you can monitor differently, you can do this. So by adding all of the other things that we know work well within certain populations of this disease, we could see the outcomes of a resolution increase from 25.2% to maybe 30 or 35% by adding that additional effect, but only if it's delivered in the right way, only if it's supported in the right way. We and the industry have battled so long to get this close. If we do not support the rollout of these medications in the right way and the right pathways, we're doing patients a disservice. And the amount of time and effort that we've put in to add that additional support to do that, we could just reap rewards in the real world. I'm expecting real world evidence to be better than the clinical trials in this disease profile.
And just think what it'll do for disease awareness. But I'll end on this note, because I know you're going to go to wrap up in a minute, Roger. A little teaser for when we do come around to the nomenclature debate. If this drug is approved for the treatment of NASH with fibrosis, will it then translate to MASH or MAFLD with fibrosis? Or do they have to go back and do another phase 3 trial? And what does this mean to all the drugs that are currently in phase 2B for a NASH indication? Do they now have to delay their phase 3 trial to go back and do a MAFLD trial? So these are some of the interesting conversations that were spun up at the nomenclature meeting, and I don't pretend to have the answers, but they are very interesting and intriguing questions to discuss.
That's a great point, Stephen. And as I've been reading some of these studies that have come up from time to time that have said if you analyze this based on NASH, you get one result, and based on MASH, you get a different result, or NAFLD and MAFLD. That's been my reaction. It's been so hard to get drugs to the finish line. Why would you do something that risked a redefinition of the work that you'd already done? It just doesn't make sense to me. I mean, if it was easy to get these drugs approved, that would be different. But as we know, it's not. So I'm wholeheartedly with you there. Okay, well, close is real simple, all right? 15 months from now. And we're sitting here because something happened with this drug. What happened? Brave one, go first.
So let's say the favorable long-term safety data has shifted the momentum of the regulators to approve obeticholic acid based on its efficacy on fibrosis resolution, while it did not show that in the cirrhotic population. And thus, we're going to have a drug in a non-cirrhotic population. Now, there was no safety signal from the cirrhotic population that was disconcerting, and we might use FXRs in a selected patient population with non-cirrhotic advanced NASH. Is one option of reading the crystal ball.
Sadly, the problem with crystal ball is you only get one read, and then everyone remembers if you blow it. But okay, so that's one. Next.
15 months from now, the field's broader than obeticholic acid, and we have a phase 3 readout on efficacy coming in Q4 from resmetirom. So again, we're just being hypothetical. So let's assume that meets an efficacy endpoint. The safety and tolerability of that drug have been reported already in 1,200 patients. We'll have the additional 900 to 1,000 from the efficacy trial. So let's say they submit a new drug application and both are approved. So in 15 months, we're looking at 2 oral therapies approved to the treatment of non-cirrhotic NASH. That would be huge for the field. That would be a tsunami for NASH. That would be unprecedented and something that all of us have been waiting for the better part, well, at least me, for the better part of 2 decades. Jorn that would take him back to his teenage years, so I'm not sure he was too concerned about NASH as a teenager. But that would be huge, unprecedented. Will that mean that we are done? No. We're not done with NASH just because those two, if we're fortunate enough to have both approved or on the market, we're just getting underway. We're really just getting underway. So what I see in 15 months is a huge groundswell of interest in the field, that needle swinging back towards research dollars coming into the space, disease awareness really, really taking off, and patients actually seeing a positive impact. Again, you're not going to cure everybody, but there will be people that we see significant reductions in liver chemistry tests, liver fat, non-invasive assessment, all things pointing to the fact that these drugs are having an impact. And obviously, it'll take a long time before we see, what that impact is. We know from hepatitis C, where cure rates are now greater than 90%, it comes down to how many of the population are treated and cured before we can begin to see a drop in liver transplantation, drops in the cost of care of this disease as it burdens our society. We still have a long way to go, but my gosh, in 15 months, if we could be there, how great that would be for the field.
And I suppose I was going to say some of those same comments, but I think for me it was the invigoration of the industry in the context that we will have OCA and yes, possibly resmetirom, and rather like boceprevir and telaprevir did for the hepatitis C world, we then get a whole host of drugs and people's interest rises and we start to go out and locate. And I think for me, and where nurses are going to fit in, it's going to be the location of this disease, it's going to be about the comorbidity management and trying to break down the silos, because we're not going to find NASH if we don't break down the silos. If we can locate early fibrosis in our biggest co-diseases, so our type 2 diabetes, our cardiovascular, then we can really make a big fat dent in this super tsunami with this. And I think we'll have too much to discuss on surfing the NASH tsunami in 15 months and then ongoing with real-world data. How exciting could that be?
As my people say, from your mouth to God's ears. Stephen, the first time I ever talked in public about this disease, the second comment I made that people remembered was the first drug is whether and the second drug is which. That you really have a market when you have two drugs. And if you're lucky enough to get two drugs with different modes of action, which is virtually never how it works, then you elevate discussion a whole bunch of levels. I was really heartened by this data. I thought, like I said, they set out to present the first thing they presented very conservatively. Here's what we had to prove. We think we proved this. There's a whole, as you point out, Stephen and Jorn and Louise, there's a whole bunch we haven't seen yet that could change our impression on that. But I'm with you. I think that this suggests that we, faster than 2025, which was what people had been thinking a little while ago, we may have two drugs with two different modes of action, both investing in education and commercialization, and that would just be massive.
Well, listen, before we close, I have to give special kudos to both Louise and Jorn. Louise for recovering from COVID and being here and being her usual chipper self, looking no worse for the wear, and Jorn for persevering a transatlantic flight only to get home and say, wait a second, kids, hold on, wife. I got to go do a podcast. I mean, I don't think we give him enough credit for we don't know what that conversation was like in the background, but thank you for being on.
Amen and amen. And Stephen, for a guy who loaded four F-250s in 109 degree, or for our European friends, 43 degree temperature yesterday, I kind of feel like I'm the only one who's done nothing special except show up. The rest of you've all been fantastic.
Yeah, it was great. And Stephen, next time I do that transatlantic, make sure you show up.
Absolutely.
So Stephen, thank you for leading us through that. That was great. Jorn, thank you for staying awake and for making compelling comments that didn't even suggest that you were tired. Louise, congratulations on recovering. We've got you off in time to go watch let's all wish Louise good luck when the English women play the Norwegian women in the Women's Euro starting in about 7 minutes. And I'll be back with the business report in a little bit.
And now for the Season 3, Episode 36 Business Report.
Slava Ukraini, Heroyam Slava, glory to Ukraine, glory to heroes, as the world keeps giving witness to this war that just doesn't seem to end. On to fatty liver disease. Last week, we left ILC 2022 in the rearview mirror, courtesy of a superb episode piloted by Scott Friedman with Neil Henderson riding alongside him. This week, we take two firm steps into the future.
To Intercept, a hearty well done.
First step, as you just heard, our primary focus is the press release that Intercept Pharmaceuticals put out last week, providing a significant update on the Regenerate trial. As we all agree on this episode, a press release cannot provide conclusive data on how regulatory agencies will respond to Intercept's new NDA filing. This well done, however, is a full-throated 3 cheers for Intercept for having the conviction to stand behind obeticholic acid after receiving a complete response letter and, in fact, doubling down by continuing to Regenerate 2 more years, expanding the trial size, and reconsidering major analytical elements. It'll be great if their efforts pay off.
And then there's the Nomenclature Conference.
As we discussed briefly at the beginning of today's episode, last Thursday and Friday in Chicago, a few hundred fatty liver stakeholders from around the world gathered to discuss what the name for this disease should be. Going all the way back to the beginning of this podcast, we've had the discussion in the background about NAFLD versus MAFLD, but it turns out the issue might be more complex than simply which of those two names makes sense. I think we're going to be hearing more about this issue as time goes by. We're not prepared to cover it yet because there's not enough clarity around it.
As to Rising Tide and CME.
We've slowed up the schedule on Rising Tide slightly to make sure we have all the promotional support elements nailed down. Stay tuned for updates. On the other hand, our CME pilot goes into production next week, and we hope into distribution the week after that. On next week's podcast, I'll provide a complete update on the episode and what makes it special.
Downloads this week are back to the levels we saw throughout May.
That means more than 1,500 total downloads the first 6 days of this week after more than 1,200 last week. We have some great topics coming up, so expect things to stay strong. And with that, I'm off. This is our first Monday recording in 3 weeks, which means Magic Mike doesn't need to do his engineering on a crazy tight schedule. He's always fantastic, but he's really outdone himself these past 3 weeks. Also, thanks to Eric, Steve, Murph the Magnificent for maintaining quality during a challenging recording and distribution schedule. And with that, I'm off. Stay safe, surf on. Look forward to seeing you next week again on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com. and we will answer on the podcast or the website.